It's a very thin layer, but it's everywhere. It covers the whole ocean.
So where the air touches the ocean, we should better know what's going on there, because a lot of CO2, for example, is passing through.
All energy and all heat and all fluxes are going to this intermediate layer.
And we want to understand the chemical and the biological processes, what it evolves into.
And within this microenvironment, you have a lot of activity, microbial activity, algae activity. So they are changing constantly. So this microenvironment can be a very strong source of CO2 to the atmosphere, or they can absorb a lot of CO2, depending on the main biogeochemical processes occurring in there.
It's so complex and there are so many open questions that still have to be answered.
So first we started with this mesocosm experiment where we kind of control everything. You don't have the influence of currents or eddies or rain or wind. So you could see the development of the slick in the sea surface microlayer. But here in the open ocean is different because you cannot control anything. So it's constantly mixing the ocean.
The SML is so close but so hard to sample. It's there, we can see it, but it's so hard to assess. We explored maybe the deep sea more than the SML before.
We all kind of know about the difficulties of sampling the SML. You have to put machinery in there, but the moment you touch it, it kind of runs through your fingers. So how do you sample it? Oliver with his catamarans brings in an option to provide a lot of SML with as little effort as possible, which is still a lot of effort, and make this phenomenon of a mediating layer on top of the ocean understandable.
So the two key players here are our two catamarans. Halobates, it's an autonomous surface vehicle, and it measures this microlayer continuously, has a lot of sensors on board. And we have Glaucus Atlanticus to collect multiple samples, which then goes to the lab for further processing.
So we are using these catamarans to sample this sea surface microlayer as far as we can away from the ship, and then take samples of this real sea surface microlayer that has not been disturbed before.
Alongside these catamarans, the BAS team are using autonomous scientific instruments called Sniffle and Bernd, as well as a host of drifters that continuously measure the surface. Our best chance to understand the sea surface microlayer is to find and study a slick, a thick, well-developed area of SML, rich in organic matter that creates a biofilm habitat for microbial communities. Understanding what's happening inside, under and around the slick can tell us how the skin of the ocean is changing our environment.
So tomorrow is our day. I'm pretty sure. I go from the best scenario. So we have the nicest slick I have ever seen, and we will go with Halobates, always crossing it. We will have Glaucus inside the slick for the sample collection. And we will have Sniffle and Bernd deployed outside the slick, and we will tow Sniffle and Bernd then inside the slick with the small boat. We will also go to Fitzroy, pick up some drifters and deploy them inside the slick. We hope with this data we can see the dynamic of the slick, how it will change with time basically. So I hope everyone is excited.
The most important thing is to see slicks.
I think it's looking very promising.
I'm really thrilled for today, yes. I really hope that this is going to be the day for slicks.
Waiting for the whole week before, like this is the condition we want to have.
So right now we don't have wind, and this is good for the slicks to develop, because then all the microorganisms in the surface, they have the time to do photosynthesis or respiration, and they can start changing the biogeochemical conditions in the surface. Then is when the slicks develop. And when you find this You are sure that something very interesting is happening there, because usually these slicks are full of bacteria or algae, and they are the primary producers of whole ecosystems.
Then we saw a slick developing around us, and I was very excited.
Really viscous and slicky, and we saw also jellyfish in there. So you can see that they support food webs, right?
And then you have to be fast, because this is a sample which is interesting now and it will not be interesting in like 5 hours. So hurry up, get the sample, bring it to the ship and try to process it as fast as possible.
It was really good to dip your glass plate into such a slick and know, okay, I'm going to filter this the whole night, but anyways, it's a good sample.
So yeah, when the catamarans come back on board and when they bring the samples, things start getting really busy. We have a plan, so it's a chaotic, organized thing, I think. It's really bustling and somebody's running around, and it's really hard to put things anywhere. So it's quite challenging, but it's still also a lot of fun and improvisation.
My personal interest is the live things in the water, so the phytoplankton and zooplankton, and that's the reason why we process them live here on board.
Because even in this bottle right now, there's stuff going on. There's zooplankton, there's phytoplankton, they're eating each other, things are being broken down, they're still photosynthesizing, so things change, and they can change fast depending on what's in there. So ideally, it's always fast, always as close to the time you collect it as possible. We grab a sample of water, immediately it was given to me in a bottle, and I take it and I put it into my instrument here, or immediately it gets filtered onto one of these filter pads. So ideally, within an hour, we have a sample, oftentimes much faster than that, in our instruments.
I thought it would be much more chaotic because there are so many groups, so many people involved, and everyone wants to do their own research. I mean, you have to bring this all in the same boat. And I think we have a good coordination, so it is working quite well.
When we did the mesocosm experiment, we were quite surprised how much more material was in the surface layer. So we found more than 10 times more particles in there as it was in the underlying water. And that was quite surprising how important and how different this topmost millimeter of the water surface actually was. This is basically the test of the results we found in the mesocosm.
Phytoplankton, so very important little small animals that are living there, and then they're producing and releasing a lot of biomolecules to the sea surface microlayer. So the composition is always different. These can have influence on the air-sea exchange. And with the whole climate change, yeah, it's just a very important topic. And there are so many open questions that still have to be answered.
So we are the Austrian part in the BUS project. And my purpose and interest here is to care for the microbes, meaning bacteria and archaea, and their function in the surface microlayer as well as in the underlying water. Here there is really a variety of people, and it's super interesting to just go around the ship and see what they measure. It gives you new ideas of what kind of thing I could build into my data set somehow.
Viruses are super abundant in the sea. They kill like 20% of ocean biomass daily, so they have a huge impact. Viruses you can't see without proper microscope, right? So yeah, I think that's pretty cool. I filter the water, do respiration rate measurements or community production, because I would like to know if the slick rather produces oxygen or we have a lot of consumption of oxygen. And then we sequence a metagenome, so we get the full genomic information of the slick, from bacteria to algae and viruses, of course. And we look for interesting genes because they tell us also a lot about how the ecosystem works.
It's a brand new experience that I really enjoy. Every time I am up there with my instruments, feeling the air on my hair, I just feel amazing. We sample every day in the pristine marine environment every time the Heinke goes out, in order to see specifically which are the aerosols present in the pristine marine environment. And what this instrument allows us is to separate the aerosols by size. The biggest sea salts that we can sample, you can see here as white, and then smaller particles that are most likely black carbon. Bringing all my stuff to make science there is really exciting. To be honest, I have never imagined I would be part of an expedition like this, and for this reason, every day working here is really, really exciting.
We're looking at the optical properties of the water and what they can tell us about the organics in the surface microlayer as well as the bulk. Looking at the particulates in the water, the phytoplankton, but also viruses, bacteria, amino acids, just everything in the water that's particles. And how it interacts with light can tell you what is going on in the water.
Rain or sunshine, day or night, wind or slicks, the BAS team members are studying the SML in as many diverse biogeochemical conditions as possible.
Since the North Sea sometimes is kind of unpredictable, we always have good and bad conditions. So I'm hoping for slicks, but I'm also hoping for rain. So maybe I'm doing sun dances and rain dances. And
So I'm actually always hunting the rain, usually on the cruise. The crew is sometimes a little bit, oh, you're always waiting for rain, we don't like it. You're the only person waiting for a storm or something like this. Precipitation is one of the most important processes on our planet, I would say. Precipitation is super important. It brings us the fresh water. We all need fresh water to live. So it's also very important to understand these processes and how these processes also change with the climate change and so on. We try to analyze the impact of rain on the sea surface, so temperature and also salinity changes, and what kind of impact different rain sizes and velocities and intensities have on the sea surface microlayer and then the underlying water, how fast it mixes with the underlying water. It's the best part to be outside, to look at those processes directly and also kind of feel it. So have the strong wind, the precipitation and so on. So I think by now they are also happy when I'm happy about the rain, and they also have a smile on their faces. So.
We also came up with very nice other ideas. So instead of waiting the whole day for slicks, we started making mixing experiments. Yes, let's do some donuts. So with the small boat, we go around the catamaran and mixing up everything. And we saw already in the data that we can look at how quick the gradients and the near surface and in the microlayer redevelops. And that is very important because breaking waves, which are a typical feature of the ocean, will disturb the microlayer. But how quick does the microlayer come back? That is a new question which came up on this cruise, basically.
These are surface drifters, and they have a GPS tracker inside. Especially in the North Sea, we also have tides, so it's not that easy to detect some slicks because they're also moving, but the drifter's also moving with the tides, so it's good because with the other measurements you really have to see it by eyes and follow them, but with the drifters you can just let do it by themselves. I really like the technology is pretty easy with this GPS tracker, but you can measure a lot. Deformation, tides, convergence and divergence processes, so if a water body is coming together or not. And we also use now some sensors on it. So we also can have a look to the first 2 meters and see with these CTDs and temperature sensors if the water is mixing or not. What we try to do is to put some of them inside the slicks and outside the slicks and compare what is happening.
Yeah, this is Sniffle. This is my baby for the cruise. Yeah, and Sniffle basically can measure gas fluxes from the ocean to the atmosphere or the other way around.
I'm a physical oceanographer, and my motivation is to understand how these currents transport heat, salt and energy through the ocean surface. And for me it's very interesting to bring all the data together to understand very small scale unexplored processes is for me, yeah, something very motivating. In the end we picked up our drifting instruments, and nearly every drifting instrument we found in a slick, and hopefully we have observed today the evolvement of the slicks in the ocean.
when you see the instrument at the end of the day? Describe what it's like.
How is it?
Like
I
Christmas.
think like Christmas. Yeah, it's nice. I think we were really happy because it's very slicky outside now.
When we processed the first slick samples from yesterday, we found really much more particles in the slick than in the underlying bulk water, so at least 10 times more, I would say, from the first look I had on the data. This has to be confirmed in the end. But this is consistent with what we found in the mesocosm, actually.
And then at the end of it, or when the cruise is over, and after a few days of a break from all the stress of the cruise, we get back together and we start to think, what did we see? How does it matter to the world, and how does it matter to the scientific community, and try to develop papers from it and then make it available to everyone. I grew up in a coastal community called Elmina in Ghana, and my master's and also my PhD has always been centered on developing codes and algorithms. But I realized that, oh no, wait, I can write codes to analyze various things that happen within the ocean. Because I think the ocean is critical to all of us. I always say I feel privileged to be part of the people studying the ocean, because this is something giving great things to humanity.
And it's a lot of work and it's really long days. But I think it's kind of always the aftermath, when you really get a chance to sit down and put all the data together, and then you can like make sense of things, and then you feel like what you did was worth it, so that you can help develop these algorithms. That it's a conglomeration of international scientists that measure different parts of the ocean all the time, and the more you put it together, the better the picture can be made. So I guess coming out here is like makes you feel good about that. Makes you feel like you're doing something to add to the general knowledge of this, because we make these models a lot of it is based on these global models that are remote sensed, and they're not really accounting for the SML. If a satellite goes over or some kind of sensor above, we can't really detect the SML directly yet. So if we can do these smaller scale measurements of this and say, okay, actually when you have this really thick SML, the carbon dioxide exchange is different. So if we can apply that into the models, we refine these models in a grand scale, I think it can help us trying to get the bigger picture of what the global oceans are doing and how they're responding to things like climate change and CO2 changes in the atmosphere. Yeah, I'm also a musician. I've been playing music all my life. What I do is take science and I put them into songs. I have songs about the evolution of algae, I have songs about waves, but the most recent song I wrote is called the SML, and it's the sea surface microlayer. We'd love to share it with you if you're interested.
And that's how we're searching for the microlayer, the sea surface microlayer, where the sky meets the ocean, a micrometer to a millimeter. Yeah, we're dipping for the microlayer. Yeah, the sea surface microlayer.
So there was a picture in the proposal where there were 2 ships, there were 2 catamarans, a few small boats, buoys, and the plane flying on top of it. And I was like, that's ambition. And when I saw it, it kind of made sense to me. Like, this was the plan. We're doing it. Like, yeah, this was amazing.
You have an idea on a round table with your colleagues, and even though it looks like very challenging to bring everything together here, I mean, that's awesome.
Woohoo! Samples! The surface of the ocean, it's covering 70% of the Earth, and we're only taking a very small part of it, just like a few liters out of those billions and billions of liters of water, to better understand what in those billions and billions of liters of water is actually happening. There's a lot of stories and chemical phenomenon happening that are yet to be discovered.